This conference will now be recorded.
Good afternoon to all. Today is Akshaya Tritiya. I wish all our delegates with prosperity of knowledge. For second session in Bheshaja Rachna Parishad, we have an eminent personality speaker, Dr. Manoj Aswarsa. Currently, he is working as senior scientist at National Toxicity Center, Pune. He has done his B.Pharm from Amravati University, M.Pharm from Nagpur University, and PhD in 2019 from Jalgaon University. He has overall 21 years total experience in academics and research. He is well skilled in short-term organ culture, microtomy, immunocytochemistry, cannulation of carotid artery. He has been principal investigator in some projects like phytochemical investigations and anti-conversion activity of Commiphora mucul, that is Google. Effects of standardized extract of Centella asiatica indica leaves on cognitive impairment in chronic mild stress in Vista rats. He has been bestowed with few awards. Some are PC Dandi Award for Best Poster Presentation at IPSCON 2015. 7th Indira Pharma ZIL 2015 for Best Poster at National Level Poster Competition. Personally, we have worked together for my PG dissertation. I appreciate his one saying. I'll always remember that whenever we are talking about research, when you are working for betterment of human, cost of research should be minimal. With this brief introduction, I request sir to start his session. Sir, please start.
Yeah, thank you, Dr. Chinmay, for such a nice and brief introduction. Uh, am I audible to all of you?
Yes,
Am
yes,
I audible? Yeah.
yes,
And the slides are visible, right?
yes, visible.
Okay. So, hello, Dr. Chinmay, please confirm whether everything is right, visibility, then my voice is getting to all of you.
Yes, yes, audible and slides
Yeah.
are also visible.
Okay. So the topic of my talk today is the preclinical drug discovery and development. Because I'm a pharmacologist, I'm a M.Pharm pharmacology. So by choice, I have chosen this preclinical drug discovery. Means what is the role of animal studies and preclinical development in the preclinical studies in the drug discovery and development, which is equally important to that of the clinical studies in the discovery of new drug. So my contact details are given over here. So if you, because most of the MD and PhD research scholars nowadays are working on animal studies for their Ayurvedic formulations and preparations also. So if you need any type of help, as Dr. Chinmay reached me long back 5 years ago, he was my he was the first collaborator from Ayurvedic background. So I start with my topic: preclinical drug discovery and development. So we all are aware that what is drug discovery and what is drug development. It is a process to bring the novel drug from laboratory to the bedside, from bench to the bedside. That is what the studies which are carried, the uh, establishment of a drug as a molecule or establishment of a substance as a drug or molecule, and after a critical evaluation of that drug, both in terms of preclinical and clinical, and after that the rigorous involvement of various scientists, chemists, biologists, pharmacologists, clinicians, and statisticians, most importantly, those who are involved in a process of drug discovery and development. Okay, so a drug discovery, it is a very long process, because a drug to be in a market or substance to be in a market as a drug, it is almost taking 10 to 15 years for the designing, for the development, and final approval of that substance as a drug to treat or to mitigate certain disease condition. As we all know, that whatever the drugs which are present in the market and whatever you people are practicing as a clinician, all these drugs are have undergone a rigorous clinical and preclinical testing. Apart from that, the synthesis or its origin. So mostly the drugs are originated from the plants, animals, or minerals or marine sources there, and mostly the chemicals synthesis in the laboratory. In the recent era, the genetic engineered drugs are nowadays are booming, and which is serving as a life saving for many critical and life threatening disorders. We all are aware of that, and we are witness to this new era of drug, and many more are in a pipeline to be in a market for the availability uh, throughout the medical practice. So this is the history of drug discovery right from 1919. In the initial period of the 20th century, the serendipitous discoveries was made, and this was the developments which are seen in the drug discovery. And in the today's era, or in last uh, I can say 4 to 5 decades, the robotics and automation and the AI and the computer based drugs and designing and all that, it makes the availability of targeted disease or targeted patients, and hope we'll have a better future for the drug discovery in future also. So coming to actual topic, the drug discovery, we all are aware what are the drug discovery, the phases of drug discovery, what are the clinical phases of discovery. I'll just highlight the clinical phases also, and afterward I'll be talking much more on the preclinical, the animals used and all that. So before a drug which is reaching to the patient, it is but obvious that it should go through the rigorous testing, and these are the steps which are involved for a substance to be approved or to be nomenclated as a drug, as a drug. So it has to go to various safety, efficacy, and uh, various pharmacokinetic and pharmacodynamic measures. So it is almost taking 15 to 20 years for a substance to be a drug in the market. So basic research in which you have to identify the target, means for what disease, for what ailment condition, disease condition, you have to prepare a drug, or you have to modify, or you have to synthesize a drug. After that, you can have the availability of lead identification and lead optimization, in which around 20,000 compounds is to be synthesized, and or whatever procured from the nature and all that, whatever we have seen in the last slide. And amongst that 5,000 to 20,000 compounds, only 250 compounds are reaching to the preclinical, because of these uh, uh, obstacles in the lead identification, lead optimization. Because whatever the eligibility of a drug or failure of the drug in the drug discovery process, very first step. Then in preclinical, 250 compounds are coming. In preclinical, again, it will take around 3 to 5 years for a complete evaluation of a drug in animals, or for the complete study of the drug in animals, in which the PKPD studies are done, the pharmacokinetics and pharmacodynamic studies, and the most important is the toxicological studies. Unless until the drug is not safe, unless until the drug is not safe, it is regardless to be to be having the efficacy. So after a thorough evaluation or toxicological evaluation, the drug will enter into the PKPD studies, and after passing that, only 5 compounds are entering into the next step of clinical studies, in which you know that phase 1, 2, 3, and 4. Rather, there are 5 different stages for the preclinical, for the clinical evaluation of the drug. And 5 compounds, amongst that 5,000, 20,000, or 250 preclinically passed compound, 5,000 enter into the clinical studies, and from that only one compound is said to be the approved drug for the post market, or post market surveillance, or the it is launched as a drug for the phase of in the phase 4. And phase 4, you know that it is lasting minimum for 2 years, minimum for 2 years, or maximum the life of the drug. The any if any type of adverse effect, any type of adverse events are reported to the drug, naturally the drug is the precautionary measures is to be taken, and it is the costliest business. More than 1 billion euros are required for a substance to be approved as a drug. why this drug development and discovery process is so lengthy, or why this drug discovery and development procedure is so costly, is because it has to pass through many steps. These are what: the drug selection or the target selection, whether it is acting on any cell or genetic makeup, whether genomics are involved, proteomics are involved, bioinformatics studies, then the lead discovery, medicinal chemistry. After lead discovery is what? It is what a synthesis of a compound, or isolation of the active molecule from the natural source. And then going for the combinatorial chemistry. Combinatorial chemistry, again, it is what? The virtual activities in which you can predict the action of that drug in a simulation manner. Then assay development and HTS, high throughput screening. It is again based on the in vitro studies evaluation, or what is the literature. Based on that, you can produce the maximum number of, or maximum synthesized compound in 10,000, say for example, if whatever is mentioned in the last slide. And afterwards, the in vitro studies are carried, and it is now rather sort of mandatory that the drug will not be evaluated directly into the animals unless until it is having some base for from the in vitro studies. So the drug have to have some in vitro studies. And in vitro is what? Which is the studies which are done apart from the body, in a petri plate, or in a cell, or in isolated compounds, or isolated tissues and all that. So it gives a basic idea about the affinity and selectivity of the drug, and whatever the cell disease model which is to be induced in the animal, that in isolated compound you can have that. And in vivo, very what common, that is in which the particular disease is to be induced in the animals. You have to check the behavioral studies. Then if needed, there is a need, then you can have the imaging studies, and ex vivo studies are done. And after that only, if these all phases are passed, which almost take 3 to 7 years up to this stage. And after that only the drug will enter into the clinical trials. And clinical trials in therapeutic, just the introduction about the clinical trials. What is clinical trial, or what are the clinical trials? These are the set of procedures in a medical research and the drug development to study the safety and efficacy of a new drug. We all are aware of that. So the trials only, I'm talking about the clinical trial, phase 0, 1, 2, 3, 4, 5. It takes almost 10 years to complete. So from lead optimization, from target selection to clinical trials, and after clinical trials, the drug which is entering into the market. So that's why I have said it is almost taking 15 to 20 years for a drug to enter into the market as a candidate for the treatment. So starting with the clinical trial, this is the very first stage. Recently have been introduced or designated as a human microdosing studies, phase 0. What is phase 0? It is a human microdosing study in which the microdosing is done. Micro means what? Whatever the drug doses you have confirmed from the preclinical studies, the subtherapeutic dose of that is given in a 10 to 15 healthy volunteers. Patients are not used over here. It is the first human trial, or first in human trial, which is basically conducted to explore or to confirm the data of preclinical studies. Okay, whatever the preclinical data we were having, to confirm that preclinical, whether it is having similar effect on the humans. At a primary level, the microdosing is done. The subtherapeutic dose is given in 10 to 15 healthy individuals, and it will simply determine whether to go or no go decisions is to be taken from phase 0. It is mostly concerned with the preliminary data on the drugs, pharmacodynamic, kinetics, and dynamics, and efficacy of the preclinical study, as I said. And after a successful completion of phase 0, the drug will enter into the phase 1. It is actually the phase first, or stage first of the clinical trial. Okay, the basic objective or aim of this phase 1 is to study the safety of the drug. Then the side effect of the drug is to select the possible dose and formulation and route of administration of the drug. So it gives a basic idea. It is again performing 20 to 100 number of healthy volunteer. Here patients are not used. Mostly it is concerned with the safety, then tolerability, PK and PD studies. These phase 1 studies are again grouped into two. That is what SAD and MAD. Many of you clinicians are maybe involved in the clinical trial. Okay, so it is very basic for them also. But it is my duty to start with the clinical trial, because unless until clinical trial phases are not discussed, then the importance of preclinical studies definitely will not be there. So this phase 1 is grouped into two, SAD and MAD. SAD is a single ascending dose and MAD is a multiple ascending dose, MAD. Okay. In case of SAD, the single dose of the drug is given, and whatever the whatever the toxic effect of that drug, or if that dose is having any type of adverse effect, it check एक single dose दिया जाता है उसमें, and if it is not showing any adverse effect, then the dose is escalated up to what? Up to a maximum concentration to which the drug can be, or the person can tolerate that dose. Maximum tolerated dose, MTD जिसको बोला जाता है. Okay, and MAD, the multiple ascending dose, it is given in multiple low doses to the patients. And the third aim of this phase is to check any type of interaction with the food, or presence of food in the stomach, whether it is affecting the absorption, or whether it is affecting any type of pharmacokinetic profile of the drug. And here again, in the phase 1, to study the food effect of the drug, the humans are divided into, or the volunteers, sorry, the healthy volunteers are divided into two groups. That is, one which are fed and one which is fasted. And then again, the absorption studies are checked in the phase 1 study. This is a phase 2 study which is performed in 50 to 300 patients. It is a controlled biological activity. It is the first stage where the apart from safety, the efficacy is to be touched. It is again done in a 50 to 300 actual patients of that disease for which the clinical trials are sought to be. So it is a controlled single blind technique. Single blind means what? Either doctor or the patients, or patients are not aware of whatever the treatment is given. It is mainly concerned with the safety, efficacy, drug toxicity, and drug interaction. Again, it is of two types: phase 2A and B. Phase 2A, it is generally performed uh, to make the base of proof of concept of the whatever the studies, whatever the results you got in the preclinical evaluation in phase 1. And phase 2B, it is responsible to determine the optimal dose for the biological activity with minimum side effect. Am I audible? Hello? Am I
Yes,
audible to
sir.
all of you? Yeah, okay. So
Yes,
phase
yes, sir.
2B, it is a dose finding stage. Then phase third, which is the most important, most critical, most expensive, and time consuming. And time consuming. Most of the drugs candidature are failed in phase 3. In phase 3, because it is a randomized control trial which is occurring at a time at various centers, multi-center, randomized controlled multi-center trial. That is what the RCT जिसको बोला जाता है, randomized control trials, which is done at different centers. Different centers means what? The different regions, maybe the geographical regions and all that, maybe the ethnicity region, centers are there, maybe some disease condition are also evaluated. It is the most expensive, as I said. Around $53 million are required for the conduct of phase 3. It is a long lasting, it is a time consuming phase. Around 300 to 3000 patients are used at each center. At each center, the study may be carried out at a time for from 10 to 100 centers, depending upon what type of drug uh, the drug is supposed to be treat to a particular disease condition. Next, it is the success chances of this phase is very less, that is 5 to 14%, again depending upon the category of the substance. It is mainly concerned with the efficacy, safety, comparison to other drugs. And lastly, if all these phase 1, 2, and 3 are passing, then the drug is entering into the phase 4, the happy stage, what you can say, in which the drug is now ready for the marketing, and the drug will remain under surveillance, and that's why it is called as the post-marketing surveillance of the drug monitoring trial phase, in which the drug have to be observed for any type of possible adverse effect or any type of any type of interactions. Yes, of course, is there. It is to be reported under the head of division called as the pharmacovigilance, in which the adverse drug reaction monitoring is done. And if a drug is having a toxic reaction, adverse reaction, if it is life threatening, then phase 4 may also result into the withdrawal of many drugs or withdrawal of drugs. And few examples I have quoted over here. You as a practitioner, you are aware of these drugs categories: statin derivative, this specific drug molecule, that is a serivastatin, then uh, troglitazone and rofecoxib, have been removed from the market after its successful introduction and completion of phase 1 to 3. फर्स्ट 3 phases उसने सक्सेसफुली कंप्लीट किया था, उसके बावजूद it was failed in the market when it was studied in some a group of patients, particularly about the coxib derivatives. That coxib derivatives were associated with the cardiac complication. That's why it has been removed from the market. Okay, coming to the original topic, the title of my topic, that is the preclinical research. So these are the stages of developing drug. Number of times I have repeated. Early drug discovery. Early drug discovery में क्या होता है कि whatever the drugs, whatever molecule which you are claiming for a drug as a drug, okay, the synthesis of that, the optimization, lead optimization, then the target and all that, all these early drug discovery is done. Afterward, the preclinical stage. Then during preclinical, if there is a success in the result, if you find efficacy and with less safety, with less toxicity, the drug can be applied, or the sponsor can apply for the INDA, investigational new drug application. Then the drug will enter into clinical research, and afterward, whatever the phases we have discussed in the last slide, approval process and post-marketing safety surveillance is done, and then the drug is ready for the consumption. So coming to the preclinical research, which is also said to be the non-clinical studies. So what basically is preclinical, or what is a preclinical trial? So preclinical trial, it is what the studies which are done in the animals, which is supposed to be, or which is hoped to be effective in humans. Whatever the trials or whatever the drug which is supposed to be there for a particular disease condition, to check that drug, to evaluate that drug into the animal, is a simple set what a preclinical trial or studies are. You know that around 50 to 100 million animals per year, ranging from simple zebrafish to the non-human primates, are used for experimentation each year. I am talking each year. 50 to 100 million animals are used on the globe. 150 Nobel Prizes, which was awarded in physiology and medicine, they have used animal models. Sorry, 180 Nobel Prizes. And this is what a data which is uh, updated till December 2021 on this link which is provided over here. You can just visit to this link. You will get a list of all Nobel Prizes right from the first inception, right from the inception of Nobel Prize, and the scientists who have used the animal species or who have used the preclinical trial for their research work, and they have been uh, what awarded or recognized with the most respectful award, Nobel Prize. Long way back, the history of use of animal models in the clinical research, it was in 5th century BC. The first for publication based on the animal studies was found to be there in 1902 by the French biologist. And afterward, the use of laboratory animals has grown year to year. And in today's era, you know that the availability of animals, the availability of transgenic animals, the modified animals, genetically modified animals, manipulated animals, and use of these animals are now, it is the rate of that use is increased for the drug discovery. Early in this 20th century, the mouse fancier began breeding of mice, and afterward, the inbred mouse in the biomedical research is tremendously increased. And this is what a beauty of animal studies, the gens, that is a genetically engineered model. Genetically engineered model means what? The animals are modified according to our requirement, according to the requirement of disease. You can prepare a rat with a diabetes inherent. You can prepare a rat with a immunodeficiency, the rat with what uh, born AIDS in AIDS or HIV infections and all that. So genetically you can modify the animals, and right now most of the research laboratories or maximum research laboratories are working on these genetically modified mice and rats and other species. This is the objective of preclinical trial. The main goal of preclinical studies is to determine a starting safe dose for the first in-human study, as I said earlier, and assess the potential toxicity of the product or the drug, which typically include new medical devices or maybe a drug or maybe prescription drug or maybe any diagnostic tool. So this is what a preclinical trial. That is what trial which is done in the animal. But why preclinical studies are done? Okay, simply to test the efficacy and safety of a drug before the drug is to be tested in the animal, and more stress is to be given on the safety. It is said, well said, that it is it is okay that drug is not having efficacy, but the drug should be safe. The candidate or the drug candidate should not have or should have the minimum toxicity, because there is no drug available on the globe without toxicity, isn't it? There is a side effect, there are several side effects, adverse effects which is to be in the drug. But the efficacy is the secondary. Primary is what a safety of a drug, and for that, the preclinical studies are done both in vitro, in vivo, and it is mandatory requirement for various regulatory authorities like FDA and MHRA. Before you are supposed to submit the IND, you should have the preclinical data. Not only the preclinical data, both in vitro, in vivo data. Okay. Apart from this, mostly the toxicological data. And most of the failure of the drug in the preclinical stage, you know that it is occurring in a toxicological evaluation, in a toxicological evaluation of the drug. And yes, preclinical studies answers to some of the questions or addresses to some of the questions which are commonly what seen or observed in a clinical trial. Very first, whether the drug will move onto the studies on human. This is what a base. Preclinical evaluation is the base for go or not to go for a drug to be used, or of the substance to be used as a drug in the human, whether to go or not. It gives a basic idea, because there is a lot of investment for the clinical evaluation of the drug. As I said, 250 preclinical, उसमें से सिर्फ 5 ड्रग जो है वो क्लिनिकल में जाते हैं, मतलब इतना एग्जॉस्टिवली वो चेक किया जाता है. It gives answer to that. What does the body do with the drug? Yes, of course, the pharmacokinetic of the drug. It gives a basic idea about the pharmacokinetic of the drug, because 95% of 95 or 99% of mouse genes which is overlapping, it is overlapping with the human ones. So not only the pharmacodynamic, but to some extent pharmacokinetic actions are also similarly seen in a mouse model, whatever we are using in the preclinical studies. It gives idea about the designing of phase 1 clinical trial. The drug is potent, but the main question is whether it is safe. जैसे हमारे आईपीसी में बोला है, इंडियन पिनल कोड में बोला है कि अगर 100 क्या बोलेंगे उसको, गुनहगार जिसको मराठी में बोलते हैं, उनको अगर वो भी छूट गए तो चलेगा, लेकिन एक भी जो गुनहगार नहीं है उनको फांसी या शिक्षा सजा नहीं मिलना चाहिए. वैसे हमारा है कि अगर 100 ड्रग्स विथ टॉक्सिक इफेक्ट but potency, it is not to be involved in the clinical trial. The drug should be safe, first criteria. Potency, secondary criteria. Of course, the drug should have the efficacy, but the primary criteria is that it should not have the toxic reaction, it should be less toxic. Okay. And lastly, it can answer to identify the criteria for evaluating safety in the humans, whether to go for clinical trials or not. It obviously detects the toxicity. It establishes the dose response estimation of pharmacology and toxic effect, assess the drug distribution to the organ system, basic idea. Then identify metabolic, kinetic, elimination pathway. I forgot here. It is mainly again to determine the dose of a possible drug. What should be the drug, what should be the dose of that drug, what should be the formulation of that drug into the which is to be entered into the clinical trial for the human. Okay, this clinical trial, the preclinical studies are giving the basic idea, and whether the drug is having any type of tumor producing capability, carcinogenic, or reproductive toxicity is done, teratogenic toxicity or potential is to be done, because these are the mandatory toxicity studies which are to be done apart from what are the main suggested toxicity studies like the acute, subacute, and chronic and subchronic toxicity. Apart from this, the carcinogenicity, reproductive toxicity, and teratogenicity is to be done. And for that, the species which is to be selected, it is mostly one rodent and one non-rodent, because whatever the study which is preclinically evaluated, it is to be done on two species minimum two species, of which one should be rodent, mostly rats, and non-rodents like dog, non-human primates like monkeys and apes can be used. Okay, so two species are required because some biological variations are seen in the rodents and non-rodents, and to to evaluate the drug completely in a preclinical, the it is to be evaluated in minimum two species. With the development, there are four main stages or categories for the preclinical development, because the drug which is entering into the clinical trial, it has to pass through several stages. You can say there's a stages. What are the developmental stages of the preclinical, which is to be done in animals like mice, rabbit, rats and mice are the most contributed species for the evaluation of the preclinical studies. So it is done in four phases. That is the safety pharmacology, very first primary, that is the pharmacological testing to check that the drug does not produce any toxic effect, may be acute, a single administration, like bronchoconstriction, dysarrhythmias, then blood pressure changes and ataxia. If any sort of this acute toxicity, means as soon as you administer the drug, a single dose study is to be done, acute study is to be done. A single dose is administered, and if there is any acute toxicity, it is to be observed in animals for 14 days, 1/4, for any type of hazardous condition like bronchoconstriction, which may be life threatening, like dysarrhythmias or blood pressure changes or ataxia. Then the preliminary toxicological testing is to be done, usually for 28 days. What we can say it as a subacute toxicity. And after 28 days, if the drug is even the drug is not showing any toxic reaction, you have to observe the behavioral pattern of a drug. You have to observe any type of changes in the fur color or reactions or leaking and all that. If any reactions are seen, you have to note it every day for 28 days. Okay, and after 28 days, you have to humanly sacrifice the animal, and post-mortem studies have to be done by taking the histopath of major organs, and by removing the blood, or by taking various, or by conducting various biochemical parameters from the blood, separating all the serum and all that. Okay. Next important step is the PKPD, that is pharmacokinetic and pharmacodynamic testing. The pharmacokinetic gives the idea about the what the body is doing with the drug, and pharmacodynamic whether the drug is having any biological effect in the system or not. And lastly, after a successful non-toxic reactions and successful pharmacokinetic and pharmacodynamic reaction, the chemical and pharmaceutical development of that drug, in what dosage form it is to be done for the large scale manufacturing or large scale synthesis and purification, to assess the stability of the product, what are the compound, what are the formulation is there, to assess the stability minimum for a year or two. Then to develop the formulation suitable for the clinical studies. Students, sorry, are you there, all of you? Dr. Chirmay, am I audible
Yes,
to all
sir.
of you? Okay, please be confirmed. Okay, so this is what statistics about the preclinical trial, the facts about the preclinical trial. You know that around 10% of the total cost which is involved for the drug in a complete establishment of the drug, complete development of the drug, 10% of cost is only spent on the preclinical studies during the drug discovery process. around 15 to 30 billion dollars are required for a substance to be as a drug complete, of which 10% means around 1.5 to 3 billion dollars are required for the preclinical evaluation. So what the thorough preclinical evaluations to be done. Approximately 80% of the compounds which is passing the pre, what you can say, the drug discovery initial steps, 80% which is coming to the preclinical trials, it get failed. 250 में से कितना 250 में से सिर्फ 5 कंपाउंड आते हैं, isn't it? Then 39% of the drugs are rejected in the preclinical studies because of poor pharmacokinetic profile. The drug, if it is having a poor pharmacokinetics, naturally it won't be having any effect. And 11% are rejected because of the toxicological issues, and that is what uh, when the research scholar come to us for the protocol, and we usually do prefer to perform at least the acute toxicological studies to them, and we suggest them to go for the toxicological study, because you are preparing the formulation, and you are preparing that formulation for the humans directly, without any base of animal studies, or without any toxicological base. It is what the base you are getting from the market only, after observing, isn't it? So because the chances of getting rejected of a drug because of the toxicity is very high, 11% is very high. So coming to the preclinical testing, the overall classification of preclinical testing is the toxicological evaluation and pharmacological evaluation. Okay, the preclinical studies require the time span of 3 to 6 years minimum. Preclinical studies, मैंने आपको पहले बताया कि it is what is performed in form of in vitro and in vivo. In vitro means what? Outside body. And in vivo means in an intact animal. So in vitro studies are giving idea about this characterization of the receptor, possible mechanism, or if there is any inhibition of enzyme, if it is having binding to a particular receptor, or if it is having some MOA, or it gives the basic about the mechanisms of action, and whether it is having any cytotoxic activity in the isolated preparations. And in vivo, vastly, that is what the is divided broadly into two, that is the toxicological studies and pharmacological studies, which together give the safety and efficacy, the dose conversion, and determination of the starting dose in the human. And for that, three steps are involved in the in vivo studies. I'll restrict my talk to the in vivo, and that too to the toxicological evaluation now. So very first step is what? The toxicological assessment of the test. As I said, the toxicity testings, toxicological evaluation of a drug is a primary objective of a drug. Toxicological assessment of the drug, in which the drug have to pass through different phases of, or different studies, like acute, subacute, subchronic, and the chronic studies. And it is simply to determine the LD50 estimation. LD50 is what? Lethal dose in 50% of the animals is used. Now, because of the OECD guidelines, which is given throughout the globe. Most of the India, of course, is the follower. Is a part of that OECD, and they have suggested to perform the chronic, subacute, and acute toxic study in animals, in which they have reduced the number of animals, and they have given certain guidelines how to perform that toxicity study in minimum number of animals with less time span. So minimum 14 day studies, maximum in chronic you can have 90, more than 90 day studies, rather 90 day studies, or maybe for a year. And after that you can whatever the guidelines suggested, you have to go for the primary evaluation of the toxicity studies. Second step, after the successful toxicity studies, means what? Non toxic report. क्योंकि अगर स्टेप वन फेल होता है तो definitely the drug will not enter into the pharmacological evaluation. But if the toxicity tests have passed, no toxic reactions are seen, then the drug will enter into the pharmacological evaluation, in which the primary and secondary pharmacological activities are done, and animal models are developed or which are already developed are used to evaluate the primary pharmacological activities of the drug. And you know that around 90% of human diseases can be reciprocated in animals, right from central nervous system to the metabolic disorder, diabetes, whatnot, AIDS, then again various infectious condition, viral infection, bacterial infection, hepatoprotection, hepatodestruction, kidney studies, heart, blood pressure, whatnot, everything, every disease which is seen in the animal, cancer, of course, all these diseases can be seen, can be induced, can be reciprocated in the animal models. And this is what the step 2, which is at most important. And if this step 2 is showing the efficacy, if non-toxic, step 1, efficacy, step 2, then the drug will next move toward the step 3. Again, the important step, that is how it is to be administered, how it is to be given to the humans. That is what the pharmacokinetic study, in which the absorption studies are done, ADME studies are done, absorption, distribution, metabolism, and excretion. Then the tissue concentration, fluid concentration, histopathological changes which are seen in the animals, it is to be done, and various biological indicators from the serum is to be evaluated for the presence of drug and its metabolite. Yeah, coming to the first portion of the in vivo studies, that is the toxicological studies. You know that toxicology is a branch of science which is dealing with the toxins and poisons and their effect, and what are the possible treatment for that poison or toxic reaction. So these toxicological studies are basically aimed to determine the NOAEL. NOAEL is what? No observed adverse effect level. मतलब ये level drug का वो level जहाँ तक वो safe हो. NOAEL is the highest dose level or concentration of the substance which defined condition of exposure causes उसको expose होने के बाद no observable or detectable side effects or adverse effects are seen. Not side effect, adverse effects are seen on morphology or any functional capability or capacity in growth, weight, lifespan, any hematological changes are seen. All these are to be studied in the toxicological study. That's why in 14 days, 28 days, 3 months, or 90 day study, or more than 90 day study, the primary aim is to study all these parameters, toxicological evaluation. Okay, this is what the three primary types of toxicological studies which is to be done: the acute toxicity, the subacute toxicity, the chronic toxicity. We'll see in detail one by one. These are the three mandatory studies to be done, and once it is found to be safe in the chronic toxicity, then it is to be evaluated along with this duration only. You can check it for the any type of teratogenicity, that is which is having the any adverse effect on the fetus, developing fetus, mutagenicity, any effect on the genetic composition or alterations are seen, any tumor producing capability the drug is having, if it is a carcinogenic or not, and whether it is affecting the immune response or it is having the ability to affect the immune response, or it is to be checked in the chronic toxicity studies. One by one we'll discuss. In short, we'll be discussing, of course, acute toxicity studies. It is to determine the short-term adverse effect of a drug when it is administered in a single dose or in a multiple dose during a period of 24 hours. Once you have administered a particular drug at a particular dose, a single dose is given, and you have to observe the animal for 24 hours. If there is any type of reactions, adverse effect, death is there within 24 hours, you have to report it. You have to report it, and you have to then report the same in again the same next set of animals. Okay, and if no death or no adverse reactions are seen, still you have to observe the animal for 14 days. For 14 days, and if unfortunate mortalities are seen with the drug, then you have to record it, or you have to what take the histopathological studies then for the evaluation of any morphological changes or pathological or any type of biochemical changes are seen. It is basically giving you the idea about the LD50 determination. This acute toxicity study may be of two types, that is a single acute toxicity and multiple dose study. This was for the first time was given in 1996 by the CDER, Central Drug Evaluation Research, which suggests a single dose acute toxicity study for a pharmaceutical substances. Okay, and you have to observe the effect for 14 days in a non-rodent and rodent species. Minimum 2 mammalian species are given. A repeated dose in which you have to repeat the dose, and the dose toxicity tested is carried out for a minimum of 28 days. 28 दिन तक आपको check करना होता है. The test substance is administered regularly at a specific time, and the usually rodent is used, rats or mice, and having the age of 5 to 6 weeks for the toxicity. Mostly the females are preferred. You can have the satellite group also. Satellite is what a group which is not actually involved in a grouping of that toxicity testing, but to study the toxicokinetic effect, how the toxic drug or how the toxic reactions are seen and how it is moving, just similar to the pharmacokinetic. That is what a satellite group in which control and high dose group is to be given and what toxic reactions are seen. Three types of acute oral toxicity studies. That is the fixed dose procedure in which a fixed dose is given, then acute toxic category ATC, and up and down in which you can vary the dose. Fixed dose में एक ही dose दिया जाता है, acute toxicity study, and then the up and down method. This is what FDP, fixed dose. This is what ATC, acute toxicity, and UDP testing. This is UDP testing. This is the toxicological testing approach most recommended by the various regulatory agencies, including India. It is what the screening method which is involving the dosing single animals and sequentially at a 48 hours interval. हर 48 hours में आपको डोज देना होता है. मतलब you can increase the dose or decrease the dose. 5, 10, 5, 50, 200 and 2000 maximum doses recommended. In UDP, the maximum dose which is to be given, the upper limit is the 2000 milligram per kg dose. In subchronic toxicity studies, which is to be done for 90 days, it is usually predicting the appropriate dose of the test substance for the coming chronic toxicity. Okay, and used to determine the no observable adverse effect levels of this toxic of these substances. This is the chronic, which is to be performed for more than 90 days, and periodically the animal have to be observed. कम से कम 3 महीने आपको देना है. More than 3 months you have to give, maybe for a year or two. The animals are observed periodically. Then the chronic toxicology and toxicology study provide inference about the long-term effect of the test substance in animals. And once you have passed the chronic toxicity study, the drug is said to be the safe. The drug is said to be the safe. Then it can enter into the pharmacological evaluation. Similarly, the carcinogenic studies is to be done. I'm going to give this PPT with the organizer. You can have the look, because this is what a carcinogen, any type of any potential of the drug to develop the tumor, of course, for the long term, for 12 months to 18 months respectively. And if any type of tumorous growth is seen with that drug, then naturally the drug will not be prepared for the efficacy studies. The toxicokinetics, similar to the pharmacokinetics, that is what the movement of the drug. Pharmacokinetics means what? Movement of a drug, and toxicokinetic means what? Movement of the drug which is said to be the poison in a pattern of higher doses of any xenobiotics or chemicals or toxins and all that. Neurotoxic studies, yes, of course, whether the drug is having any type of adverse effect on the brain or brain function, neuropathological lesions and neurological dysfunctions like loss of memory, sensory defects, or learning and memory dysfunctions are seen, and for that various behavioral animal models are set. All type of CNS disorders can be induced in the animal, right from epilepsy to the anxiety or depression also. though it is not termed as a depression in case of animals, what we have defined in case of humans. But yes, the similar symptom, depression like effect, anxiety like effects can be induced in the animals. And if any drug is having toxicity to the what brain or neurotoxic, if it is neurotoxic or if it is causing dysfunction of the neurological functions, then that is to be evaluated. Okay, the test substance may be administered for 28 days or even for 98 days, and after completion of that 90 days, you have to observe the any type of neurological changes or neuropathological changes. Coming to the in vivo preclinical testing. Okay, in vivo, the very first step after passing the toxicological test, the drug is coming into the in vivo clinical trials. In vivo, the screening of the drug is to be done. It is a simple and rapid performing the test to indicate the presence or absence of a particular activity in a drug. Very simple. Simply, you can induce the, just for example, I'm telling, you can induce the pyrexia in the animal, and you can have a check on the antipyretic effect of your test drug. You can induce the inflammation, you can induce the algesia, you can induce the asthmatic condition, you can induce the tuberculosis or whatnot, whatever the disease, and estimation of or evaluation of your protective evaluation of protective effect of your test drug. That is what is screening. Isolated organs, yes, isolated organ. For example, I give you a very simple example of trachea. Trachea of guinea pigs are generally used for the evaluation of antihistaminergic activity. Histamine, in a similar way, whatever the action of histamine which is seen in the humans, the same sort of actions is to be observed in case of animals, and guinea pigs are more sensitive amongst this. Antihistaminergic, antisecretory, vasodilator activity can be well studied in the isolated tissue. Even for the undergraduate practicals, we usually use the isolated organ preparation to study the cholinergic effect, anticholinergic effect, effect of various salts, effect of histamine, effect of again adrenaline on some isolated tissue organs and all. For that, ileum, trachea, then fundus, anacoccygeal muscle. All these muscles can be used to give the primary basic idea about the in vivo, or what you can say the in vitro evaluation of the drug, to have a basic in vivo training or training trials. Then bacterial cultures to study the effect of substances as antimicrobial, whether it is having any antibacterial effect. Antibiotics can be generated. Yes, as I said, animal models. You just name the disease condition, we can induce that disease condition in the animals. Just example I have given over here, the kindled seizure in the rats. The seizure, similar sort of kindles. Seizures are induced in the animals. The whatever is seen in the humans. Petit mal, then the large epilepsy or the petit mal and grand mal epilepsy, both sort of epilepsy can be induced, and for that, typical models are developed. For example, if I give the example of seizure, there is a compound called as a pentylene tetrazole, PTZ, induced seizures in the animals. Or you can have the genetically hypersensitive rats, experimental tuberculosis in mouse, though the laboratories are not permitted for induction of tuberculosis. Those who are in the facilities can induce the tuberculosis in the animals. And after that, the general observation, the drug is injected in the triple dose to the small group of animals, and it is observed for any type of overt effect, whatever the expected effect are there, depending upon whatever the disease or whatever the induction you are supposed to be there. The study will prolong for 2 days to 2 months or 6 months even. This in vivo preclinical data, it gives the mechanism, possible mechanism of action of the drug. Just for example, your drug is having the antihypertensive effect. Okay, but how it is having the antihypertensive, whether it is alpha blocker, whether it is a beta blocker, whether it is inhibiting the S, or it is having the CCB or calcium channel blocking action. So these animal models specifically to block the alpha receptor, to block the beta receptor, to block the S, or to inhibit the S, and to block the calcium channels can be induced in animals. So mechanistically, you can also evaluate the blood pressure. Yes, blood pressure can also be evaluated in the rats and mice with a specific instrument available. In next in vivo preclinical trial is the systemic pharmacology, that is irrespective of the primary action of the drug, its effect on major organ system like nervous, cardio, whatever the vital organs we can say. Uh, the systemic toxicity, systemic pharmacological actions are to be tested. The quantitative test, the DRC, the drug response relationship, the maximum effect. Even not necessary, you have to always use the rodents and rats and mice, and because nowadays for undergraduates during your MBBS programs also, the rats and mice are totally banned for the use. But you can have the alternative of using the Chickilium isolated preparation for the primary demonstration of experiments to the undergraduate people. Okay, you can have the goat tracheal chain. We have the established model for that. Goat tracheal chain, Chickilium. We can have earthworms for the evaluation of anthelmintic activities and all that. So all these are what I mean to say. The preclinical studies are equally important in a nutshell, and after that the confirmatory tests are to be done. Okay, so coming to the animals which are used, the types of animals which are used in the result in the research, mostly mice and rats are contributing around 95% of the total animals which are used in the research. As far as the mammals are concerned, I am talking, and remaining this 5% it is contributed to this, the guinea pigs, then the rabbits, hamsters, farm animals. Cattle are also used. Horses, you know it very well for vaccine developments and all that. Cats, dogs, non-human primates, and other fishes, mollusks, and all that are used. But the maximum contribution or maximum mice and rats are the most frequently usually used as a mammals in the rodent category. Okay, just to mention the names and uh, to make you aware about the animals, I have compiled this list. The mouse, the rat, the rabbit, cat, dog, guinea pig, pig, primates, frog, and fish, mollusks, insects are used. Mouse most frequently used. Rats are most frequently used. For what? For the pharmacological genetics of mammals, virology, models of human disease, etc., etc., etc. Rabbits, of course, it is used for pyrogen quantification. Nowadays it is replaced. Then again for the cosmetic evaluation, but now it is totally banned since 2004. Then cat, now restricted. It is used for the evaluation of CNS and respiratory system before the development of these rats and rabbits and genetically modified animals. You can have the well versed Alzheimer rat, the neurodegenerative rat, the SCID rat. Then you can have a particular depressed condition. You can induce the depressed condition in the animals. That's why the use of cats and dogs nowadays are very restricted. But yes, our guide and our teachers have worked well on the cat and dog. The beagle, it was used rather. And most of the laboratories, again, the research institutions are using a dog for the electrophysiological and neurophysiological evaluation of the drug. Yes, we are using guinea pigs frequently. Guinea pigs are the most sensitive species for histaminergic and what are the pollens which is there in the environment. Mostly it is used for the evaluation of certain substances or devices for the auditory system also. Pigs, it is used for the training purpose. Then primates, rhesus monkeys, of course rhesus monkeys and all these primates, non-human primates, are not the part of academic institution, but yes, for the research institution they are, it is well available. Frogs nowadays are, it is totally banned, but as a undergraduate student or postgraduate students, we have sacrificed many frog. We have studied number of drugs on the cardiac parameters, on the electrophysiological parameters, on the NMJs, neuromuscular junction studies in frogs we have used. Fish, mollusks, and insects nowadays are emerging one. Fish are very interesting one. Yes, as I said, these are the two main species which are used of rat. That is a Vista rat and SD rat. SD rat is a Sprague rat. It is depending. The name is given based on the uh, resources of that uh, animals. The Vista rat and Sprague Dolly rat. You people might have heard this two name, and it is the most frequently used rats, mostly the Vista rats. But you can modify the animal according to our need, according to the disease. For example, these are some examples I have quoted over here. The biobreeded rat, it is what a nod non-obese diabetic rat means. What it is, having a type 1 diabetes by born. You have to manipulate the drug, or sorry, you have to manipulate the rats, and you have to prepare the rat which is by born diabetes, specifically to evaluate the drugs acting on type 1 diabetes. Type 1 diabetes, even you can induce type 1 diabetes, type 2 diabetes in the SD rats or in the Vista rats, also in the mice also. Then this second is long Evans rat, which is for the evaluation of behavioral and obesity research. Long and Evans have researched out that. Then Zucker rat, you can see it here, for obesity and hypertension. It can grow up to 1 kg. Usually the weight of a rat is, what we are using for the preclinical evaluation is from 125 to 150 to 250 grams. But in case of the Zucker rat, it can grow up to 1 kg. Okay. Hairless for immuno compromised studies. Then RCS, that is the Royal College of Surgeons, for the retinal degeneration. SRK, not Shah Rukh Khan. SRK, it is a shaking rat Kawasaki, that is lowered expression of renin protein. The renin protein, the deficiency of renin protein, it leads to the Alzheimer disease or epilepsy and autism. So you can lower that expression of the protein, and you can develop the animal models for that. These are some examples. Long Evans rat, then the Zucker rat, biobreeding rat, and hairless rat, whatever is shown in the last figure. Just for information, I am giving you the basic about the mice. These are the mice. C57BL6 mice we are usually using for the immunity and infectious condition, whereas this Swiss albino matlab white, it's a condition. Swiss albino mice are most commonly used for various evaluation, for various toxicological evaluation, terratological evaluation, for various biases, for the evaluation of analgesic activity, anticonvulsant activity, anti-inflammatory activity too, because it is a small, easy to handle, cheap, very cheap. This, at a cost of 200, you will get a single mice, and 500 for a rat you have to pay. So as compared to the clinical, and as compared to the other studies or other models to be used, the rats and mice are the cheapest one, and the quantity of the drug which is required is less. These are the advantages of the rats and mice which is used. This is a guinea pig. These are various experimental uses of guinea pigs, mostly the related to the respiratory bronchodilators, anaphylaxis condition, histamine, antihistaminic, digital biases, etc. etc. This is very docile. This is very docile. You might have seen the guinea pig. Most of the families are having the guinea pig as a pet also. It is very docile, very obedient, very cooperative. जैसे आप बोलोगे वैसा करेगा वो. It is what the animal, that is guinea pig, was used in the discovery of vitamin C and development of vaccine for the diphtheria, tuberculosis, and for the evaluation of local anesthetics. This is jabil, though not frequently used in educational institution. Hamster, the third commonest species which is used after rats and mice. Then again, as I said, SCID, that is a severe combined immunodeficient mice. What you can see over here, it is a compromised animal. It is a immunocompromised animal, and if for the evaluation of anti-HIV drug, the SCID mice can be used. This nod mice, by born it is a diabetic, mostly type 1 diabetes. It is a non-obese one, and this is what a zebrafish nowadays. Since last 2 decades, the zebrafish is emerging as a one of the most common screening tool as an animal for the preclinical evaluation. what is the Danio radio, okay, for the evaluation of various genetic functions and genetic evaluation of the drug. Last slide, that is, of course, whatever animals we are using, whatever preclinical evaluation, rodents, non-rodents we are using, we have to have the ethical regulations and laws for the control of use of animals, for the to avoid the cruelty to the animals, because before even 1990, 1990 2000 तक भी CPC-SEA, which is a committee in India to look after the control and supervision of experiments on animals, okay, which was actually the CPCC was framed in 1964, but practically after 2000 only the CPCSEA actually started working in the academic institution as well as the industrial institution. So very first law to avoid or to prevent the cruelty to the animals by the central government of India. I have compiled this list from India only. Okay, it was the Cruelty to Prevention of Cruelty to Animals Act (PCA Act) जिसको बोला जाता है, which was framed in 1960 and amended in 1982. And afterward, the CPCSA, still the institution, those who are having the animal facilities have to have the approvals from this committee, CPCSA. It is a committee for the purpose of control and supervision of experiments on animals. See, this CPCSEA, it is not restricting you to use the animal, but they are supposed to look after the proper use of animal. Wherever there is a need, use minimum number of animal, use whatever the procedure you are following, what anesthetic to be given, which procedure is to be followed for the euthanasia, what are the recommended anesthetics, and what is the dose, what not to be done with the animals, what surgical procedure is not to be done to the animals. These all are framed in the guidelines of the CPCSEA. There is a phobia amongst the student and among the faculties of many medical organization or medical research institution that CPCSE is banning the use of animal. No, they are not banning. They are not restricting the use of animal. But yes, of course, the proper use of animals they are recommending. Okay, after that only various against Wildlife Protection Act, you know it very well, INSA, then the Breeding and Experiment of Animals, that is Control and Supervision 1998 acts. Everyone is not having the right to breed and sell the animal. You have to take a separate permission for your animal house to have the breeding license and all that. ICMR guidelines are given, then your MCI guidelines was given, where they have restricted the use, where they have not recommended the use of animal for any undergraduate course, not for BMS, not for MBBS, dental, pharmacy, or any allied medical substances. No animals are permitted even to demonstrate for the undergrad programs. And recently in 2012, the UGC have framed the guidelines that the discontinuation of the dissection of animals experimentation in zoology or licenses in the phased manner unless until it is required for some research purpose. Okay, so this is all about my presentation, ethics and animal use. This is what the facility which is available at our place, and because I have seen most of the proud Ayurveda Acharyas are nowadays involved in research studies, in the animal research studies, and very proud of them that you people are involved in the authentication of your formulation by means of using the animals. So to approach and to know the facilities which are available at our place and what studies we are doing, what instruments we are having with us. This is what a compilation. This is the mission and vision of our institute, and these are the approvals. Our lab is FDA approved. Of course, it is registered with the CPCSEA as per the norms. We are having the license to import export code. We can import the animals as per requirement if there is a need. We are having the ISO certification for biological evaluation, and this ISO certification is also for the testing for medical devices. And most importantly, the DSIR and Zero, it is generally given to the NGOs and trust. And we, as a trust, Apt Research Foundation is a trust, is a foundation which is having the permission, which is having the approval certification from DSIR and Zero. DSIR is the Department of Scientific and Industrial Research, and Zero is the Scientific and Industrial Research Organization. And after having these approvals, we can import the animals without the custom duty, and the GST is exempted. Only 5% GST we have to pay as an approved institution from the DSIR and Zero. These are the animals which are available with us. Mostly the small animals, it is approved, of course. The CPCSC is not supposed to approve for the large animal house in the academic institution or research institution. We are having all these species of animals, right from white rabbits to SD rats, then Vista rats, guinea pigs, C57BL mice. And yes, for the students and those who are interested, for the research scholars, uh, we are organizing a training every year or as per the requirement about the handling of the animals and animal cell culture also. So if here in a week day training or 10-day training, depending upon the demand, we are providing the platform to you people to handle the animal, to give the basic about the animal. We are showing the dissection of the animals, video organ isolation, histopathological, how to take the slides and all that, and even the animal cell cultures. These are the facilities of instruments available with us. Almost all behavioral instrument as well as the biochemical estimation. I'm not going to read all the list. Histopathological, then ELISA reader. This is what I was talking about. 8-channel data acquisition system power lab. It is used for the determination of blood pressure in animals. That is NIBP, non-invasive blood pressure, in which similar to our human, a cup is tied over the tail, at the end of the tail, and you can have the blood pressure measurement, you can have the electro cardiography, ECG, heart rate, SBP, IBP, and etc, etc. All automated. Biochemistry analyzer, hematological analyzer, urine analyzer is available with us. These are the animal studies we have conducted so far, all behavioral, right from hepatotoxicity to wound healing, antioxidant, biocompatibility studies, MTT assays, cancer, anti-cancer drugs, and all that. And these are our collaborators. Nowadays, most of the Ayurvedic colleges from Maharashtra and many regions of India, North India, are approaching for their evaluation. We are having a approved PhD center from the MUHS and Symbiosis International University also, SPP also, Pune University. We are having collaboration with renowned universities like NMIMS, Indus Biotech, Agarkar, IIT, Powai, etc. Okay. Last but not the least, this is very beautiful. Whatever medications or medicines or medical procedure we have undergone, if you have undergone any medical process or medicine, you have benefited from the animal testing. So a big salute to these animals for giving their lives to evaluate the drugs and to make the substance available as a drug for the humans. So big thank you for your kind listening, and big thank you to this cute species. Thank you very much. Yes, I'm done with my presentation, Dr. Chinmay.
Sir, uh,
Yeah,
it was very nice session.
thank you.
And if,
My contact details are given. Let me share first.
okay,
You might have noted it down. My contact details are given on the first slide. You can, we can have a personal interaction through mail, or my phone numbers are also given. Both the numbers are given. you can have a personal contact with me. Is it visible?
Sir, it is
Yes, numbers
visible.
are visible. Okay. And if you miss it, you can take it from Dr. Chinmay. I'm ready to take two, three
Participant
questions. And
having
if you
any
want to
question?
discuss, yeah, if you want to discuss anything, if you want any type of guidance in terms of animal experimentation and animal studies, uh, I'll be very happy for you people to contact me anytime. You can mail me. Dr. Shridini, I can see Dr. Shridini is there.
Yes, madam is also there.
Okay, okay, Chinmay, then I think the topic
Yeah,
was not.
we'll end up with the session. If any question is there, I'll forward to you.
Yes, please, sir. No problem.
Yeah. Thank you for this scientific session. It was very much explanatory.
Thank you.
Thank you.
Thank you. So, should I stop the display sharing?